Great. Welcome, everyone, to the American Society of Gene and Cell Therapy's briefing on gene editing technologies, transforming rare disease care. My name is Margarita Valdez-Martinez. It is a pleasure to welcome ASGCT's president, Dr. Jeffrey Chamberlain, who will kick off today's briefing with an overview of what we'll be discussing during our various presentations. Dr. Chamberlain?
Thank you. Hi, I'm Jeff Chamberlain. I'm the current president of the American Society for Gene and Cell Therapy, and I'm a professor at the University of Washington, where my own laboratory studies gene therapy for the muscular dystrophies. If I could have the next slide, please. I'd like to kick off by sharing a little bit about the American Society for Gene and Cell Therapy. We're a professional association that's comprised of more than 6,000 researchers, scientists, physicians, patient advocates, industry professionals, and many others that are working to advance the cell and gene therapies. Our organization was founded almost 30 years ago, and most of our members are scientists, with about half of them based in academia and the other half in biotechnology and pharmaceutical companies. Next slide, please. I just wanted to highlight that one of the most exciting developments in the gene therapy space over the past year was the approval in December of Casgevy, a combined cell and gene therapy for sickle cell disease. Casgevy uses the new method of gene editing using CRISPR-Cas9 to modify a patient's own blood stem cells, which are then infused back into the patient. It's also used for a related disorder called beta thalassemia, and it was developed by CRISPR Therapeutics and Vertex, who brought it to market. A second gene therapy for sickle cell disease was also recently approved. It's known as Lyfgenia, and this method uses direct gene transfer into a patient's own blood cells and is marketed by Bluebird Therapeutics. And we've got great hope that many more approvals are on the horizon. I wanted to point out that sickle cell disease affects nearly 100,000 Americans, mostly Black, and we think it may turn out to be the most far-reaching gene therapy approved to date. Next slide, please. So I'd like to now turn to the existing pipeline of gene therapies. Every quarter, the American Society of Gene and Cell Therapy releases a landscape report that updates the field on the universe of gene and cell therapies that are approved throughout the world, but also that exist in the clinical pipeline. And the next slide shows the latest data that we've accumulated. Today, there are more than 30 gene therapies that have been approved throughout the world, and about half of those are currently approved in the United States. There's also 29 RNA-mediated therapies that have been approved worldwide and 65 non-genetically modified cell therapies. For years, the biotech, pharma, and healthcare world have been discussing the advent of gene therapy as something that is coming soon. And while the patient populations may remain somewhat small right now, the age of gene therapy is clearly already here. Next slide, please. As you can see, as of January 1st of this year, there are almost 4,000 gene therapies in the pipeline that are being developed. More than 2,100 of these are gene therapies. There's also an additional 900 non-genetically modified cell therapies and more than 800 RNA therapies. Next slide, please. And when we zoom out on all the different gene therapies under development, you can see that about half of them are being brought along for cancer indications. And the left part of this slide, maybe a little hard to see, is showing the preclinical data. In other words, gene and cell therapies under development, and the drugs that are already in the clinic are listed on the right. So rare diseases comprise the majority of non-cancer targets, but there's many other targets that are being developed against disorders of muscle diseases, brain, heart disease, and many other indications. And as I mentioned, gene therapy is well on its way to becoming routine medicine. So I'd now like to move on to introduce some of our more distinguished speakers, the first of whom will be Dr. Fyodor Urnov from the University of California, Berkeley.
Thank you, Dr. Chamberlain. Folks, my name is Fyodor Urnov. I am a professor of molecular therapeutics at the University of California, Berkeley, and I'm also a scientific director at its Innovative Genomics Institute, which was founded by Dr. Jennifer Doudna, who together with Dr. Emmanuel Charpentier won the Nobel Prize for inventing a method with gene editing with CRISPR. So I really want to thank the leadership of the American Society of Cell and Gene Therapy and my colleagues for the opportunity to take just a couple of minutes to explain how gene editing with CRISPR works. It's a technology that can be explained in accessible terms, and understanding how it functions will help appreciate its potential for genetic cures. Next slide, please. Gene editing, changes human DNA inside living cells of a person. So the challenge it has to solve is our genome is very long. It's in fact a meter long. So if you read the whole thing, one letter a second, it'll take you a century to read the whole genome. Now, I will explain in a second how CRISPR gene editing accomplishes the amazing feat of changing human DNA precisely. But first, let me offer a 30-second preview of what Dr. Chen and then Dr. Porteus will speak to, which is genetic disease. Next. It's a sad fact that even a single typo in this century-long text can cause severe disease. And shown here is just one such typo, with the letter A replaced with the letter T, in a gene that makes a protein that produces hemoglobin that carries oxygen in our cells. And this mutation causes a devastating disease, which Dr. Chamberlain just mentioned, sickle cell disease. It affects more than 100,000 Americans, millions worldwide. And as Dr. Chamberlain just said, there is a CRISPR cure for it. And you'll hear shortly about this from Dr. Chen and Dr. Porteus. My job is to take us back to the fundamental science of what CRISPR does. And next slide, please. Before I do that, I want to mention that gene editing is every technology. It stands on the shoulders of giants. And in our case, it's an earlier form of using DNA as medicine. It's called gene therapy. I need to briefly tell you about it to provide some context for why editing is exciting. Gene therapy is used to treat and in some cases cure genetic diseases. It works by taking a virus that normally infects human beings, removing its viral genes as much as we can, and replacing them with a gene that replaces the one mutant in a given disease. So for example, the medicine Zolgensma is used to treat spinal muscular atrophy. So the replacement gene fixes the one that's broken in folks who have the disease. So this therapy works. It's being administered directly to young children or even newborns with this disease. But there is unfortunately some evidence that its benefits wane over time. You see the virus washes out as the baby grows. Another gene therapy, next slide please, called Libmeldy for metachromatic adrenoleukodystrophy, uses a different virus that inserts itself into blood stem cells. Now, the topic of cost is not something we're going to be discussing much today, but I do want to mention that this medicine was just approved and it costs more than $4 million per child. In part, and this is the relevant bit, because it's very expensive to make this virus. Next slide. I'm going to do a metaphor here, and I hope you will forgive me. But I think this is accurate. If you have a car with a flat tire, gene therapy is taking the spare, adding it somewhere onto the car, and hoping it runs. And as I mentioned, this actually works. Gene editing is repairing the flat. Let me walk you through this. Next slide. Dr. Chamberlain just mentioned Casgevy, a heroic and historic development for our field, a treatment using CRISPR-Cas9 gene editing for sickle cell disease. So Casgevy for sickle cell disease, as any CRISPR medicine, has two parts. It has a protein part with a rather bland name, Cas9. It does the editing. But then remarkably, and this is the Nobel Prize winning work, it has a nucleic acid part, which is evocatively called a guide RNA, which guides or leads this editor to the right gene. How does that technically work? Let me tell you. Next slide. So imagine you want to target a gene inside human cells with CRISPR. You take Cas9 and you arm it with a location code to find the right gene. And when Cas9 finds it, it will drive the editing process. Let's look at individual letters of the genetic code. Here's a gene you wish to edit, and there in red is the letter you want to fix. The way to do that is you take Cas9, the editor, and then you pick 20 letters in your target gene, right where the change needs to be evoked. You then take just the RNA, and you put inside the RNA a 20-letter match to that gene sequence you want to target. And the rules of making this match is something that we study all in high school. A equals T and C equals G. Once you've done that, you send CRISPR inside the cells, and Mother Nature takes its course. 3 billion years of evolution are behind it, where CRISPR-Cas9 finds the target gene and creates a chemical change at the target that ultimately drives gene editing. Remarkably, the gene editor then goes away, but the genetic change persists. Next slide, please. I took this slide from the biotechnology company Intellia. And they showcase the fact that CRISPR is quite versatile in terms of the genetic outcomes or the molecular outcomes of the target gene. You can use CRISPR to knock out a gene. That's been done in the clinic. You can use CRISPR to repair a gene. That's being done in the clinic. You can use CRISPR to even insert a gene, and such clinical trials are on their way. Next slide. I want to close my section by pointing out that these fundamental genetic rules of how you arm CRISPR to target gene 1 versus gene 2 have a remarkable scientific implication for how you go from making a medicine for one disease from making a medicine to a different disease. I'm going to stay in my lane of talking about mechanism, but I do want to evoke some recent clinical trial data from, again, the company Intellia, because I think they showcase this remarkable feature of CRISPR of being able to make a new medicine with relatively simple changes most robustly. So here we have the human DNA, our familiar chromosomes, and the biotechnology slide in a company Intellia, click, went after a disease called TTR amyloidosis. That's not the important part, but the important part, click, is that the gene causing the disease is on the 20th chromosome. And so Intellia used these rules to build a match in CRISPR to that gene, and they put it into human beings, and click, they showed pretty remarkable efficiency of lowering that toxic gene level protein output in the serum of these human beings. Okay, now what? Now they decided to go after a different disease, hereditary angioedema, which is caused by a mutation in a different gene, click, which is on the 4th chromosome in a completely different place in this century-long bit of text. And guess what, folks? The way they made that other medicine, CRISPR-Cas9 is the same. The framework of the guide RNA is the same. All they did, the delivery is the same. They just changed 20 letters. And then they put that medicine in for a completely different group of people with a different disease, and click, the efficiency of getting rid of that gene was just as high. What a remarkable technology. I will now pass the floor over to Dr. Janice Chen, who will walk you through the implications of this for the gene editing pipeline.
Thank you, Dr. Urnov. My name is Janice Chen, and I'm the co-founder and chief technology officer of Mammoth Biosciences, a biotech company harnessing ultra-compact CRISPR systems for in vivo gene editing. The topic of my section today is going to be on the overview of the gene editing pipeline. Next slide. You've now heard about how CRISPR can edit DNA from Dr. Urnov, but in order for CRISPR to work as a medicine, the editing machinery must reach the target cells and tissues. There are two general categories of CRISPR-based medicines. The first is called ex vivo editing, where gene editing takes place outside the body. As you've heard from Dr. Chamberlain's introduction, the world's first CRISPR-based medicine is an ex vivo therapy, which involves extracting stem cells, editing them in a lab, and then infusing the edited cells back into the patient's body. Although this process is very complex, for instance, it requires bespoke manufacturing, requires a patient to undergo chemotherapy, and the entire treatment journey can take up to a year, ex vivo cell therapies have paved the way for using CRISPR to create functional cures for certain blood disorders and blood cancers. The second category is called in vivo editing, where gene editing takes place inside the body. Unlike blood, cells in other parts of the body, like muscle and brain, cannot be extracted, so editing has to occur inside the body. In vivo editing can be delivered locally to a specific organ or systemically through intravenous delivery, a process that would be much simpler and more cost-effective than the ex vivo editing process. In fact, we envision a future where the patient experience could be similar to current practice with monoclonal antibodies, in which patients receive an in vivo gene editing medicine with a simple IV infusion in an outpatient center and then simply head home with a permanent cure after treatment. But there's still technical challenges with in vivo editing because it needs the help of a delivery vehicle. There are two types of clinically validated delivery technologies that can carry CRISPR cargo, which include non-viral methods that target the liver, such as LNPs, the technology used to deliver mRNA for COVID vaccines, and viral methods that target tissues beyond the liver, like muscle, heart, brain, or lungs, such as AAVs, which are non-pathogenic viruses that have been modified to deliver DNA to target cells. Although regulators have not yet approved any in vivo gene editing therapies, it's considered the future of CRISPR and how gene editing could address a broad range of diseases and reach many patients. Next slide. In just over 10 years, the CRISPR field has evolved from a biochemical proof of concept to the world's first CRISPR-based therapy. This figure summarizes current gene editing programs by stage and tissue type based on publicly available information from private and public gene editing companies. Each circle is representative of the relative programs in each phase of development. In the next section, you'll hear from Dr. Porteus, who will elaborate on the different phases of the clinical trial process. Today, the field is progressing rapidly with many different types of gene editors being developed and several programs advancing to the clinic or currently in clinical trials. But as you can see, the absolute number of programs moving through the gene editing pipeline is currently small and focused only on a handful of diseases in blood and liver. So why is this the case? Well, first, the gene editing field has been focused on starting with severe diseases with greatest unmet medical need to establish safety and efficacy for a new technology. Second, from a technical standpoint, certain diseases are more amenable to gene editing due to disease biology, delivery, and maturity of the editing technology. For instance, the genetic basis needs to be known and the disease biology should be well understood and tractable. There also needs to be validated delivery technologies that can deliver the gene editing components to the right cells. And of course, the editing technology has to be mature enough to generate a therapeutically relevant modification. Blood and liver are the most amenable targets because of the relative maturity of the biology, delivery, and editing technology, which is reflected in their later stage of development in the gene editing pipeline. In comparison, most programs targeting tissues beyond the liver, as indicated with the light purple circles, have not advanced beyond research. Specifically, programs targeting the brain, muscle, lung, and ear are largely untapped areas in part because of these technical challenges. What we haven't shown here are all the innovations at the top of the funnel. In other words, research done primarily at academic or nonprofit institutions, which often rely on government and philanthropic funding and fuels a substantial portion of the downstream translation and development of transformative medicines. Next slide. From these clinical trials, the current estimate is that approximately 200 patients have received or are currently receiving some form of experimental CRISPR therapy. But despite the advances in gene editing, there's still over 5,000 genetic diseases with no available cure. So how do we go from hundreds to millions of patients cured of genetic disease? We can think of the opportunity across two axes, unlocking gene editing from rare to common diseases and enabling broad patient access to gene editing. Today, the most advanced treatments in the gene editing pipeline are in rare diseases like ATTR amyloidosis and hereditary angioedema. But there are still thousands of rare diseases not currently being addressed, a challenge that Dr. Raymond will elaborate on more in her section on rare disease. Diseases that affect a significant portion of the population, such as cardiovascular disease and diabetes, are moving into clinical trials, and recent articles shown here highlight stories of some of these brave patients that are undergoing experimental in vivo CRISPR therapies. These early clinical trials are essential to establish the foundation for gene editing to address the underlying genetic causes of more common diseases. Even for the approved sickle cell disease and transfusion-dependent thalassemia therapy, access and affordability hinder widespread use of this drug for the hundreds of thousands of patients in the US alone who suffer from these diseases. But we must recognize that we are still in the early days of CRISPR, and safety and efficacy must be established before going broader. Our vision is for gene editing technology to be ultimately democratized, analogous to the widespread use of computers today. Think of the first computers, which cost tens of millions in today's dollars, where programming was a slow and difficult task and machines took up entire rooms. Today, it's an afterthought that we walk around with supercomputers in our pockets that cost only a few $100s. The promise of CRISPR as a programmable platform means that developing the components to treat the first diseases accelerates the development of gene editing therapies in adjacent tissues or indications. To help achieve this vision, advances in technology, infrastructure, and policy will unlock the massive potential for gene editing. This could involve advancing in vivo editing technologies, creation of common resources to accelerate and scale manufacturing, and forward-looking regulatory policies and incentives to address both rare diseases as well as access for broad populations. Next slide. Finally, the gene editing pipeline extends beyond therapies, and here are just a few more examples of how CRISPR will reshape our future across many industries. In research, gene editing tools have given scientists more precise methods to probe the fundamental biology of how cells and organisms function, which could unlock the next breakthrough discovery. In healthcare, we talked about its impact in therapeutics, but CRISPR is also enabling advances in diagnostics, vaccines, and identifying new drug targets. In agriculture, precision editing is also making advances in generating crops with disease resistance, improved crop yields, and increased nutritional value. And finally, with the ability to fundamentally change the code of life, CRISPR must be used ethically and responsibly, as the possibilities for gene editing can seem limitless. With that, I'd like to thank you for your attention and pass it on to Dr. Porteus.
Hello, my name is Matthew Porteus. I'm a professor of pediatrics and stem cell biology at Stanford University. I've also been fortunate enough, I'm a physician scientist, and I treat patients with pediatric hematologic diseases and patients undergoing stem cell transplantation. I was also fortunate enough to be involved in the early development phases of Casgevy, which, as you've heard now, is the first commercially approved drug using CRISPR-based technology. What I want to take us through, and I want to thank Drs. Chamberlain, Urnov, and Chen for their excellent introduction to this next stage, which is how does a drug get developed and what's the patient experience as they for gene editing based drugs. So next slide, please. So the standard and traditional clinical trial process is that there are extensive preclinical studies done in a laboratory that support the submission to the FDA of what we call an investigational new drug application, or IND. This IND has to show preclinical evidence of safety, has to describe the risk benefit of the potential therapy, and has to describe whether there's potential efficacy. Once the FDA clears the IND, which doesn't mean that the FDA gives a stamp of approval, it's simply saying that they have met the bar to initiate a phase 1 trial, which is a safety trial. And this is usually testing a small molecule or a drug or a device in a small number of healthy volunteers. And if it's proven to be safe or to have a degree of safety that justifies it moving on to a phase 2 trial, it is then tested for whether this drug or intervention would have efficacy in several hundred patients with the disease. If there is a signal of efficacy, then the next stage is to go into a phase 3 trial in which thousands of patients might be treated to determine whether the efficacy meets or exceeds currently available therapies. And if it passes that, the drug can then get FDA approval and become a commercialized drug. The fourth phase of drug development is what's called the phase 4 stage, which is post-marketing monitoring of safety and efficacy in case that there are unexpected side effects that don't show up in the first of 3 phases of the clinical trial. This is particularly important because often clinical trials are not focused on a broad and diverse population, and unanticipated side effects may only be identified when a more diverse population is treated. And this is why the FDA and it's California Institute of Regenerative Medicine and many other institutions or entities are now mandating that investigators assure that their clinical trials include a diverse population so that the safety and efficacy can be determined in those populations. Now, next slide, please. Yeah. In genetic medicines, it's a very different approach. It has the same fundamental structure, but because these rare diseases are rare, there's a different approach to them. So if we go to the next slide. So again, preclinical studies have to be performed on the safety and efficacy of a gene therapy or gene-edited drug, including animal studies. And again, a IND has to be cleared by the FDA. But instead of the trial first being tested in healthy people for safety, the first trial can be done in a small number of people with the disease. And the primary goal of this phase 1/2 trial is, of course, to measure the safety. But these patients can then also be measured for whether the drug has some efficacy. If the drug, gene-edited or gene therapy drug, has safety and efficacy in this phase 1/2 trial, it's usually in a small number of patients, often a handful or perhaps 10 to 15 patients. Those results, then the sponsor of the trial, that is the entity that is leading the drug development process, will then meet with the FDA to have conversations about what a phase 3 registration trial would look like, discussing the number of patients that might be needed and what are the endpoints that, if they are found, if the drug hits those endpoints, would justify approval. And so this is an incredibly important conversation about how to design the registration trial. So again, often it's not a comparison trial, because for many of the diseases for which gene-edited drugs are being developed, there is no baseline therapy to compare to. So you're simply looking to design measurements that this will work. And if it does pass, and as Casgevy did pass, it gets FDA approval. And again, these drugs need to be then monitored in the larger population as they're given to more patients. Now, so that's the drug development process. What about the patient experience? If we go to the next slide. So this is the current landscape of the centers in the United States that are approved to administer Casgevy. And what you can see is it's, a handful, maybe 2 handfuls of centers around the country. But this is even in the drug development process, the number of centers can be even smaller than this. So what does that mean for the patient experience? What that means is if you're a patient with sickle cell disease who might live in the Pacific Northwest or North Dakota, you don't go to your local center to get to the drug, to receive the new Casgevy drug. Instead, you have to find the time and resources to get to a local center, for example, in Southern California. So what this means is that not only is the cost of the drug perhaps limiting, but the patients and their families' ability to travel, take time off work, to take time off school to get to one of these treatment centers can be a barrier to access. And that's certainly, as I said, even more true in clinical trials where the number of centers might be half as many as this. But why do we need to do this? Well, these are complicated therapies that have some known side effects, you need to be at a center of excellence to take care of, but they also have unknown side effects where it's important that the patient is monitored by experts so that unknown subtle side effects might be noticed. So the patient experience and access to these therapies is very different than, say, a standard drug molecule or biologic, which can be administered locally. Instead, patients will have to travel to get to them. Finally, I'll just say, and before I pass it on to Dr. Raymond, is that as you may be aware, our healthcare system is fragmented. So if a patient is covered by Medicaid in Wyoming, but needs to receive therapy in California, there are barriers to getting Wyoming Medicaid to subsidize and pay for treatment in California. And so this is and now the White House and other entities are working including ASGCT, are working to try to develop a streamlined system so that this barrier is minimized. But currently it exists as a major barrier in terms of getting patients enrolled from out of state. So with that, I will turn it over to Amy Raymond to describe the importance of gene editing for rare diseases.
Thank you so much, Dr. Porteus. Hi, everyone. I'm Amy Raymond, Executive Director, Therapeutic Strategy Lead for Cellular and Genetic Medicines at Worldwide Clinical Trials. We are a contract research organization executing these clinical trials alongside on behalf of our drug development partners around the world. Next slide, please. Many rare disease communities are really looking to gene editing as a potential source of hope for much needed and durable treatment solutions. We've talked a little bit about rare diseases, but I'd like to take a couple of steps back and set the stage, define a few terms. Here in the US, the Food and Drug Administration defines a rare disease as one affecting fewer than 200,000 people in America. It's worth noting that roughly half of these patients are children, many of whom will perish before their 5th birthdays. While each rare disease is in fact rare, there are many, many rare diseases. So consequently, there are roughly 30 million Americans living today with a rare disease. That's approximately 1 in 10 people, or 1 person in the last crowded elevator you happen to be in. If they were to form a human chain, the US rare disease patients would wrap around the world 1.5 times. That is an awful lot of people. Rare diseases can be found in every therapeutic area, usually some of the most challenging development programs in each of those therapeutic areas. Shown there on the right, a beautiful pictorial representation from the FDA 2020 Rare Disease Day. Next slide, please. By most recent calculations, there are over 10,000 rare diseases. Not every one of these rare diseases, however, affects 200,000 people in the United States. You heard Dr. Chamberlain describe the recent approval of Casgevy at the top of the hour, noting that this new treatment for sickle cell disease has the potential to be one of the most far-reaching gene therapies to date, given that there are roughly 100,000 sickle cell disease patients in our country. Certainly, there are rare diseases which do affect up to 200,000 people. But there are also rare diseases affecting just a handful of people. And all of these rare disease patients, regardless of the size of their patient community, deserve a chance at a happy, healthy life. This was the basis of the Orphan Drug Act passed by Congress in 1983, aimed at stimulating the development of safe and effective treatments for rare diseases. Incentives include tax credits for a portion of the research and development expenses incurred by the drug developer, waivers for some of the typical approval fees, which can be hefty, and extended market exclusivity. Next slide, please. The human impact of 30 million Americans facing a rare disease is staggering, but so is the economic impact. In 2019, the Government Accountability Office estimated the cost of rare diseases to be the better part of $1 trillion in that year. That public report dovetails to the Economic Burden of Rare Diseases in America report by the EveryLife Foundation for Rare Diseases, which took a prevalence-based approach to derive the national economic burden by patient age and type of rare disease. These costs can be broken down into direct costs, indirect costs, and non-medical and uncovered costs. For a rare disease family with a pediatric patient, that added up to over $60,000 per year extra costs to them, and over $80,000 per year for families that have an adult rare disease patient. Next slide, please. There are untold number of smart, passionate, dedicated, driven researchers working very hard to understand the mechanisms of these rare diseases. But despite these best efforts to date, for the past 40 years, only about 5% of rare diseases have a single FDA-approved treatment. I think we would all agree that an FDA-approved treatment, which by definition, as Dr. Porteus just said, has been proven safe and effective in a well-designed trial, those approved treatments are very much a step in the right direction, but rarely is that treatment everything that's needed to bring a full relief to the rare disease patient. For example, we now have multiple FDA-approved treatments for the devastating disease of ALS, also called Lou Gehrig's disease in the United States. But no combination of these approved drugs returns the patient to the life any of us would want if it were our father, our brother, our husband. Nonetheless, they're all tools we should have available in the clinic, and each one represents incremental progress beyond what was available to the ALS community prior to their approval. As I joined you a few moments ago, I said that there are many rare disease patient communities looking to gene editing as a source for hope, and that's because roughly 80% of these 10,000 rare diseases are the result of a genetic defect. Many, but by no means all, of them being mutations that could conceivably be solved with gene editing. Gene editing treatments offer the potential to address rare diseases at the source, hopefully leading to meaningful change and durable solutions for these patients. Next slide, please. I know we have several rare disease patient advocates with us today. These are the people who are truly best positioned to speak for the thoughts and concerns of their respective communities. But based on my experience with rare disease gene therapy trials over the last many years, in addition to ongoing conversations I have with rare disease patient advocates across all therapeutic areas, I can say there are some core concerns that come up with any gene therapy trial. as well as an additional layer of concerns that need to be taken into account for gene editing products. Some of those more common examples are on the left. How will my family get to the clinical trial site and afford to stay there as needed? How will I continue to participate once I'm home? One additional question is, will I be eligible for future clinical trials? But when we layer in the gene editing, additional questions come up like, will my employer find out I received this? Will I still be eligible to receive treatment if it didn't work or work as well as we had hoped? How will we know if unintended changes were made to my DNA? Will those changes be passed to my children or to my children's children? And also, who will be responsible for my care if there are unintended changes with negative consequences? These are all factors to be worked out with data through the clinical trial process, but issues on our minds. Thank you.
Great. Thank you to our wonderful speakers. We're now going to open up the conversation for Q&A. So if you have questions of the speakers, please feel welcome to drop your questions in the Q&A chat. You can also submit them anonymously if you feel more comfortable in doing so. And then, Caitlin, I'll ask you to make this, to be able to display all of the speakers. If you can take down the Q&A slide and show the full profile for the speakers, that would be great. Thank you so much. So the questions are coming in. I did have a few questions that I wrote down while we were listening to the presentations. Dr. Chen, you talked a lot about the applications for gene editing for rare diseases, but you also touched a little bit on the application for more common diseases, especially with in vivo treatments, for example, like lung and brain disease. Can you expand a little bit more on that and give maybe a high-level overview of how the treatments would be different for someone receiving gene editing for brain disease or lung disease versus doing ex vivo?
Thanks, Margarita. That's a really great question, and I think an important distinction between the ex vivo therapies and what the potential of in vivo gene editing therapies could look like. When we're talking, currently we have clinical trials for diseases in cardiovascular disease, for instance, targeting a gene called PCSK9, which is a protein that's generally produced in the liver. And as I mentioned, a part of the challenge for effectively delivering in vivo therapies is having the right delivery vehicle that can take those gene editing components to the right tissues and organs where the editing effect will have a therapeutic output. In the case of PCSK9 as a target for cardiovascular disease, the concept is similar to how Fyodor described, where you can bring that CRISPR component to the liver and knock out this gene that will remove this PCSK9 protein, which gives folks a high likelihood of developing cardiovascular disease in their lifetime. So it's a very elegant process, which involves three main components: delivery, the gene editor, and then the guide RNA that targets that particular gene. But the exciting part of that is that it can be delivered systemically, so through an IV injection, where that medicine is able to then target that particular area where you can actually get the therapeutic effect. This is obviously very different from the ex vivo therapies that we know of today with Casgevy that requires extensive treatment that requires, extracting those cells, editing them separately, and then reinfusing them back into the patient. But as we think about other tissues and organs like brain and lung, I think those are where we're going to see a lot more effort, but also a lot more challenges because currently delivery, especially to the brain, is a very challenging process. There's a lot of limitations in terms of crossing the blood-brain barrier, as an example, and being able to deliver those gene editing components to some of these more complex tissues is something that's actively under research. But there's early indications that it can be done, but it will certainly take some more time for that research to mature.
Thank you. Much appreciated. Actually, I have a question for Dr. Porteus as well. You talked about clinical trials and you laid out really elegantly the difference between clinical trials for the standard clinical trial process versus the clinical trial process for cell and gene therapies. And, in your notes also noted that for cell and gene therapies, oftentimes the clinical trial population is much smaller. And you touched a little bit about being able to see efficacy in cell and gene therapy trials. Can you expand on that and the difference in when we see efficacy for cell and gene therapy trials versus for more traditional trials? You're on mute, Dr. Porteus.
Yeah, sorry about that. Yeah, and another excellent question, and really two parts to it. At least two parts to it. One is the issue of these being rare diseases, and so what that means again is it requires then a lot of conversations between the sponsor of the trial and the regulators, because there simply is not enough patients to do a comparison trial or to do a randomized trial. And so that's the nature of it. What I didn't mention, but I'll also say is in a more traditional clinical trial, the patients who are treated or the people who are treated in one phase don't get rolled up into the next phase. Whereas in a gene therapy or gene editing clinical trial, the patients who are enrolled on the phase 1/2 portion of the trial can get rolled into the phase 3 registration trial so that you don't need to double your numbers. And this is essential. Otherwise, you would never get an approval for a rare disease. There's simply not enough patients. Now, the reason you can have that conversation with the FDA between the phase 2 and the registration trial is because you often get efficacy. These are not drugs where, as I said, you first test them in healthy people and then look for efficacy in patients with the disease. So even in your first few even in your first few patients, you can start to measure if there's efficacy and the degree, the extent of efficacy. I think Dr. Raymond pointed out that sometimes, and even with gene therapy drugs, they can show efficacy, but maybe they're not 100% perfect. So there's still room for improvement. But it is that number of percent of patients who improved and the magnitude of their improvement that gives statistics that allow the sponsor and the FDA to then determine how many more patients need to be treated to get approval. Finally, just why do we see efficacy? Well, it goes back to what I think all of us have referred to, which is that early preclinical work, not just about testing whether the gene editor might work, but the often decades of work of human genetics, human physiology that help you understand the relationship between the genetic disease, the genetic cause of the disease in the DNA, and the genetic manifestations of the disease in the patient. So that in sickle cell disease, we know that if you have this single nucleotide change in the 6.6 billion letters of the genome, as Dr. Urnov described, that results in red blood cells having certain features. And what you can see in the very first patients is those features start to disappear. You start to see the markers, the biomarkers of the disease disappear, even in the very first patient treated. And that's a very different sort of scenario than we see with other more complicated diseases.
Thank you, Dr. Porteus. The next question that we received, I'm actually going to send this one to Dr. Raymond. For clinical trial patients, and you talked a lot about the pediatric patient population, for the patients and also for their parents, do they seem eager to enroll? What are some of their concerns or hesitancies? And how do you counsel them through that clinical trial process?
Absolutely. I would say that the perception of the opportunity is really a function of what is the value proposition for that community, right? For example, earlier we heard Dr. Urnov tell the story of Zolgensma. I was very fortunate to be part of that development program years ago, where those children we knew without a doubt would perish by age 2. So the value is, can you change that storyline for your newborn child? They are eager to see a difference for their children, that is for sure. I would say by and large that is a trend, is we're willing to move mountains for our children. Adults tend not to be as eager to jump in there, but overall this industry is really looking to use this technology in cases where we have the highest unmet need, where it is the most dire consequence if we don't bring a solution forward.
If I could add, in my personal experience, I found that patients and their families, because they have to live with the disease every day and all of its manifestations, are often more excited about new therapies than even their own doctors, and certainly than the regulators. The daily, hourly, by minute impact these diseases really, I think, motivate a large number of these patients and their families. The lived experience is really important.
Could not agree more. And when it is an inherited rare disease, one that you know you've passed on to your children already, that's another factor that plays into the value proposition for any prospective patient. You know, if I participate today, will it make the potential for a treatment for my child?
Absolutely. And when you talk about, and this is for the entire group, but piggybacking off of Dr. Raymond and Dr. Porteus, when you talk about the lived experience and the patient experience, you know, one thing that comes to mind is for Luxturna, for example, not gene editing, but just an example, which for the audience, this was developed to treat rare eye disease, which can often cause blindness. And during the clinical trial process, it was noted that with treatment that the patients were able to then walk in dim light without bumping or falling. So kind of pivoting that example to gene editing, are there patient experiences that you've noted or seen that maybe aren't traditionally captured in other clinical trials? We're like, this is something that we should consider for a gene editing therapy.
Maybe I'll take a first pass at that. I mentioned that before any gene editing or gene therapy enters a clinical trial, there is extensive study of the relationship between the genetic basis and the manifestations of the disease. So one goes into the clinical trial having a sense of what markers and what symptoms one should be monitoring. But I think what you've highlighted with Luxturna is that sometimes during the trial, you realize that the markers or the measures that you thought were going to be relevant aren't as relevant as you thought, because your patients are telling you, look, I can see, I can walk. And so you have to develop new assays and new measures. And again, that's where the investigators, the clinicians, the parents, the participants, all have to be flexible in their thinking, but so do the regulators. They have to have an adaptability that a measure that might have worked 30 years ago for a certain small molecule is maybe no longer the appropriate measure when you have this therapy that addresses the disease at its root cause and perhaps can address it for have lifetime durability. So flexibility on thinking about what are the approvable endpoints that we should be really looking at that are meaningful to patients, not just meaningful to a sponsor.
Great point, Matt. And I just want to chime in there and say, you know, luckily, our regulators have required that we include patient experience data as part of this complete package that they entertain for, so should something be moved forward to commercialization. Increasingly, I see the use of video, which I think is phenomenal. To be able to literally see the difference for these participants before and after treatment. Sometimes there isn't a great way to capture it quantitatively prior to that trial. Some developers, for example, Spark, that Luxturna story, develop brand new ways of measuring it along the way, which I think is fantastic. But sometimes the difference is something you can see quite well and makes a big difference.
Great. Thank you, Dr. Raymond, for highlighting that regulators, FDA, use the patient experience data. And since this is a congressional briefing, I do have to give a nod to Congress for mandating that FDA do that. And, you know, as a whole, I know the field is looking at ways to meaningfully include the patient experience. And as you mentioned, Dr. Porteus, and many of the other panelists today, is maybe developing new markers, maybe developing new ways of looking at how the patient is receiving the therapy. So I know that we are getting close to the 1 p.m. Eastern time. Before we wrap up, I do want to give every panelist an opportunity to go around and give your closing remarks on what you're excited about, what you see coming down the pipeline, any points that you'd like to reiterate and you really want to drive home. I'd like to give you this opportunity. And I will start in the order of operations. I'll start with you, Dr. Chamberlain.
Okay. Well, I appreciate all the folks that are listening today and, taking into account what we're saying. And, it's been tremendously helpful having Congress step in and help out, set the regulations and mandates to the FDA to help keep this going. All I want to say is that the gene therapy field is rapidly expanding. I think it's going to have a profound impact on modern medicine. It's going to reach the lives of millions of people. And, the best is yet to come. We're getting going. We're learning every day, and the technologies are amazing. And just can't be more excited about the future of medicine. So, thank you.
Folks, I just want to say one thing again, staying in my lane of science. We humans as a species have a tendency of having ideas before they become real. Jules Verne proposed in 1868 to shoot people onto the moon out of a cannon. And then it took 100 years before Americans walked on the moon. And the reason I bring this up is the dream of human genetic engineering to treat disease is 50 years old. And the first genetic engineering of humans in the clinic was in 1989. We took a long time as a community to get to a point where, as Dr. Chen, Dr. Porteus described, we can change a single letter in human DNA in a human being with a curative outcome. And the reason I connected it with the spaceflight, with the moonshot, is like any technology, it's not going to just spontaneously blossom and yield its fruit across every realm of healthcare, as Dr. Raymond alluded to. Dr. Chamberlain is exactly right. The technologies of gene therapy, the early stage clinical trial data are absolutely magnificent, but I really want to turn to the legislator to appreciate the fact that the true impact of CRISPR gene editing and gene therapy more broadly is not driven by technology alone or clinical trial data. Deploying that robustly and scalably in the context of American healthcare is a huge challenge that is not solely on the shoulders of the physician, scientists like myself, physician scientists like Dr. Porteus, or anybody else. Ultimately, it has to be a sort of nationwide affair. So on some level, I'm advocating for a CRISPR cures moonshot of the 21st century as being a major goal that we as a nation can achieve to bring these curative outcomes to patients.
Well said. I like to remind folks that breakthrough technologies like CRISPR also didn't come out of nowhere. In fact, it started from really basic scientific curiosity. And just like to remind this audience and anyone who listens that we really need to continue funding that basic research, because the next CRISPR really could be in those early academic research labs and early research. So just wanted to remind folks the importance of that basic research funding.
Yeah, let me echo what Dr. Chen said. Absolutely, that's the big part of the funnel, and we need to make that even bigger. But I will go down the funnel a little bit and say that there's a lot of really exciting proof of concept studies that are being done in laboratories, whether it's in a tissue culture dish, whether it's an animal, and we still have a challenge in getting those studies out of the lab and into patients in these early phase trials. There is only so much one can learn from a mouse. There's actually only so much one can learn from a dog or a monkey or a pig. And ultimately, the bravery of the patients who volunteer in these phase 1-2 trials should not be underappreciated. I mean, we honor them every day. On the other hand, there are many, I would say, therapies that right now are still stuck in the lab because of the amount of resources that are needed to get them out of the lab and into patients. And so we still need to come up with some creative solutions to make that part of the funnel even bigger.
Well said, Dr. Porteus. I suppose in closing, I would like to thank ASGCT as well as this expert panel, in addition to everybody who has invested their time into learning about this with us today. But one final thought from me would be my gratitude to all of those families, as Dr. Porteus said, who volunteer themselves, their time to be part of this. even bigger than that, the communities who partner with drug developers, because all of the challenges being pointed to, those are real. But one of the ways that once we get to the development stage, we can help optimize things is to have that collaboration between the community who is going to be receiving this potential treatment and the drug developers, because I think that's where we start to look at things like what is most important, how do we measure it? How can we make these things most operationalizable? How do we pre-solve the challenges that plague every development program? So thank you.
Thank you.
Fantastic. Thank you to all the panelists for lending your time and expertise. Your knowledge in the field of gene editing cannot be overstated. So this really is an immense benefit, and we appreciate your time. Also want to thank all of the congressional staffers and patient advocates who were able to join today. We will be available via email if you have any follow-up questions. And I'd also like to encourage you to look at ASGCT's website and check out our patient education page and information on our clinical trial finder. If there's any way that we can be of assistance to you, your family, or constituents that you represent, happy to help. Again, thank you, everyone, and have a great afternoon.
Thanks so much.
Thank you.
Bye-bye.